Today I'm going to be talking about how a mutation causing the addition or deletion of one base pair results in the production of a truncated or shortened non-functional mutant protein. We can use paper clips as a visual for the amino acid chains. Here are the normal and mutant amino acid sequences. Now, the reason the mutated sequence stops after histidine is because there's actually a stop codon there at the end, and knowing this will help us figure out the coding sequence of the mRNA. Now we need to look at all the possible codons that could code for the normal and mutant sequences in order to determine whether the mutation that occurred was an addition or a deletion. Now that we see all the codons that could code for the amino acids, we need to look at where the two sequences first start to differ. We see that the sequences both start with methionine and tryptophan and then start to differ at the third amino acid. Since both possible codons for phenylalanine start with U, but all the possible codons for isoleucine start with A, we can determine that the mutation occurring here is causing the addition of the adenine between the guanine and uracil, throwing off the rest of the amino acid sequence in the mutant by one base. Now we need to continue to look at the codons to determine the coding sequence of the normal and mutant mRNA strands. When we get to phenylalanine and isoleucine, we can see that both the codons for phenylalanine starts with two U's, and the only codon for isoleucine with two U's following the A is AUU. Now the only codons for histidine start with C's. This means that the codon for phenylalanine is UUC. Next, we see that all the codons for threonine starts with AC, which means the codon for histidine has to be CAC. Next, we see that all the possible stop codons start with a U, meaning that the threonine codon has to be ACU. Now we see that both the codons for lysine start with two A's, so the stop codon for the mutant must be UAA. Lastly, the codons for lysine and glycine could be any of those, since it can't be determined using the mutant protein anymore because that amino acid sequence ended with a stop codon. Here's the complete coding sequence of the mRNA that codes for the mutant and normal proteins side by side so you can get a better look along with our paperclip amino acid chain.